Good morning, everyone. Today we are starting a new series for the GATE. The first topic is enzyme. From here, we usually get 2 to 3 questions. So let's start with the enzymes. So all we know is enzyme is a biocatalyst which fast forward the chemical reactions or biochemical reactions. So the enzymes these are basically proteins, and sometimes they are uh, RNA also. RNA means a catalytic RNA that are usually known as ribozymes. So enzymes are proteins and RNA. Here we know that there are some enzymes which don't require any other chemical component for its activity, but there are some enzymes that require a chemical component for its activity. These chemical components, these are known as cofactors or coenzymes. So these components are cofactor or coenzymes. Mainly cofactors are inorganic molecules and coenzymes are generally uh, organic compound or inorganic compound, but they are in the form of compound. Now, these cofactor or coenzymes are specifically classified into another group based upon its binding to the enzymes. Means, if a cofactor or coenzymes tightly bonds with enzyme, or we can say it binds with the help of covalent bond, then this is known as prosthetic group. So we can say that a prosthetic group is a coenzyme or cofactor or metal ions that is covalently bond with the enzyme. Now, we know that some enzymes require chemical compounds and some don't. So, when we taking the general example, so we usually see that there is a requirement of chemical compound or iron, metal ion or some compounds for its activity. And the basic example is DNA polymerase, which usually require magnesium ion for its activity. In PCR reaction, we generally see it as a essential component. So, enzyme is structurally divided into two parts. One is known as apoenzyme. This apoenzyme is only the protein portion of enzyme. Protein part, it is known as apoenzyme or apoprotein. So, this apoenzyme is the protein part, only the protein part. Now, the second uh, thing is holoenzyme. Holoenzyme is the combination of this protein part and coenzyme or cofactor, which is generally a non-protein portion. So here we can see that a protein part is inactive, uh, means the apoenzymes are generally inactive as these enzymes require a coenzyme or cofactor for its activity. So the apoenzyme part is inactive. Holoenzyme is a whole enzyme, means it is having a protein portion and a cofactor or coenzyme portion, that is non-protein portion. This whole is active, means this is a active enzyme which can work and perform its activity. The holoenzymes are active and apoenzymes are inactive. So here we can look for something that is essential for the enzyme, that is its catalysis. So we are going to see how enzyme catalyzes its reaction. So in general terms, we say that enzymes lower the activation energy of reaction, that is the required amount of energy needed for a reaction to occur. They do this by binding to substrate and holding it in a way that allows the reaction to happen more efficiently. Means when we generally say this is an enzyme, okay, here a substrate bond. So for its activity, there this compounds or substrate require some collision theory. We have that in a perfect collision, which is which is directed precisely, is thought to be proceeded forward to be transferred to product. Means जब ये हमारा सबस्ट्रेट है उसको एक ऐसे ओरिएंटेशन में रखते हैं जब दो मॉलिक्यूल का कोलिजन हो तो प्रोडक्ट जल्दी बने। और ये कोलिजन इसलिए ज़रूरी है क्योंकि ये एनर्जी प्रोवाइड करता है। सो दिस इज बेस्ड ऑन कोलिजन थ्योरी और समथिंग लाइक दैट वी कैन से that we have to manage a proper orientation and collision for the substrate to be converted to product. Okay, but here when we talk about enzyme catalysis, this is what an enzyme does to substrate for the catalysis. But what it is actually doing is it is minimizing the activation energy. Activation energy is also known as energy barrier that the substrate had to cross for converting to the product. Here you can see that in this chart that here it is a substrate, this is a product, and this is showing energy, and this is reaction progress, means the binding and uh, bond forming and bond cleavage reaction that is going to be that is happening to the substrate. Here the enzyme does is this is the original required energy for a reaction to proceed. Okay, this is the top, which is known as transition state. So what enzyme does is it lowers the activation energy. So if a enzyme is involved in the reaction, then the energy required will be here. This much energy is required if an enzyme is there, and this much energy is required when the enzyme is not present. So when we see it in terms of energy, free energy, every compound have its free energy that is denoted by G. So S, which is substrate, in its ground state it have an energy uh, that is denoted by G, and same goes for P in its ground state. Now what enzyme does is it lowers the activation energy or energy barrier. So this is the energy required for a substrate, or this much energy is required for a substrate for crossing or for proceeding forward to the product. Means when this much energy is given, then only can a substrate can be converted to product. So this transition state, this is like a plateau. This is showing that when an molecule gains or presents in transition state, it can either be converted to substrate or either product. So we know that the enzyme does not affect the equilibrium, means it does not favor any type of reaction like forward reaction or backward reaction. It does not affect the equilibrium constant that is generally denoted by KEQ. Okay, so enzyme does not affect that, and I have also shown it by transition state. Now repeating it in a simple word, this activation energy or any energy barrier, consider it as a wall. Okay, and here is substrate. When a substrate gains enough energy, so that it comes to the transition state, means this peak of the wall, when substrate is standing here in the peak of the wall, it can either jump to this side or it can either jump to this side. Okay, so this is like when it is jumping backward, then it is converting into substrate itself, and when it is jumping forward, then it is converting into product. So this is a simple analogy, and from here we can say that enzyme affect rate of reaction, not equilibrium. Means what enzyme does is it fast forward the reaction, means reaction rate it increases. And how it increases reaction rate? By minimizing the energy required for the reaction. Okay, so it does not affect G. We know that ground state has its own free energy, and uh, for substrate it has its own G, and for product it has its own G, means free energy. So it does not affect the substrate and product. So we can say that enzyme only affects reaction rate and does not affect the free energy or equilibrium of the reaction. So here are some questions. You can pause it and try to solve. I will be directly solving these questions. So the first question is, which one of the following statement is correct for enzyme catalyzed reaction? Del G gives free energy and KEQ is equilibrium constant. So we know that enzyme does not affect G, means free energy, or Ke. So the answer will be D. Correct. Okay. Now next question. A protein without its prosthetic group is known as. So we know that an enzyme have a protein part, okay, and a non-protein part, and combining this we get a complex that is known as holoenzyme. This is non-protein part, this is protein part, this whole is called holoenzyme, and this protein part only is called as apoenzyme or apoprotein. So the answer is option 1. Okay. Here is another question, that is, which of the following type of molecule act as biocatalyst? So a biocatalyst is basically an enzyme. Okay, and we know that an enzyme is a protein or an catalytic RNA. So it can be RNA also. Phospholipid, we don't see it. So this is not a phospholipid. It can only be a protein or RNA. So the answer is P and Q only. This is the classification of enzyme and its general reaction. We need it in some places, so it is basically we have to remember it. There is no other option. So I hope that you can remember the following, and thank you for listening to the lecture.